Hello everybody, this is the circulatory system, the second half regarding blood vessel. This is the first video on this. Earlier in the class we talked about blood and heart, and so today I'll talk about the vascular system with respect to the circulatory system. This involves arteries which carry blood away from the heart, veins which carry blood back to the heart, and capillaries, which is kind of in between the two where gas exchange happens. I'm not going to talk about lymphatic system really at all today. And to be honest with you, I will discuss primarily arteries and a little bit of veins, and particularly with respect to differences in vertebrates and with evolution. Just to point out that a couple terms: vascular or vaso means blood vessel, and pulmonary referring to lungs. So there are a number of similarities in blood flow between different vertebrate groups. If you can follow my pointer over here, you've got fish. And you notice fish are very simplistic. There's a heart, and then you've got kind of blood vessels leaving the heart, and this is a single circuit kind of system where it goes out to the body and comes back as sort of one loop. Whereas amphibians, reptiles, and mammals that are shown here, the heart pumps out as a dual circuit to lungs and back, and then another pump sends it out to the body and back. You can see how amphibians, reptiles, and mammals are kind of increasingly more complicated with some of these things. Some of the similarities you see between all of these is, first of all, is kind of obvious. There's a heart, but the heart always pumps out on the ventral side, and that's something you can't see from a 2D picture. It pumps out ventral. And it pumps into either a ventral aorta or systemic arteries, and it's always pumping in the cranial direction. It's going toward the head. So we can't really see because there's no heads on here, but it's going toward the head every time. Here it's going toward the head. All the blood vessels kind of go up and toward the head every time. Everything goes toward the head, toward the cranial direction. Then, in all cases, there's some kind of arch. And over here, you can see in fish, you see how it makes an arch, and it turns direction over here, changes direction from going cranial to going caudal. And now after the arch, you've got stuff going down these arteries where it's going toward the caudal or toward the posterior. Same thing here. Even a little bit in the pulmonaries going to the lungs, but particularly in the systemic, there's an arch here and there's an arch on this side, and now they're going from cranial to caudal, or from anterior to posterior. They're changing direction. So we see that at all times. The other thing, which again isn't apparent on 2D, but when it changes direction, going from cranial to caudal, it goes from ventral to dorsal. So ventral being kind of toward the belly side, toward the front, in our case toward our chest, and then when it goes over this arch in these aortas, these aortic arches, now the blood is flowing kind of closer to our backs. So it's flowing kind of on the dorsal side, near kind of just in front of the spinal cord. It's also important to note that no matter what species you're talking about, this dorsal aorta, it's always rich in oxygen, or richer anyway than the veins. There's a little bit of a mixture here, but it's rich in oxygen, especially compared to the veins. And in all cases, there's some kind of systemic capillaries, and then veins that are coming back to the heart from the system are always going to be kind of poor in oxygen through what they either called cardinal veins coming back or through vena cava. The last thing, and it's not shown here, has to do with how the brain is getting oxygen. So coming off of sort of these arches over here, it's kind of coming off over here off of the dorsal aorta, and then if you think about any of these tetrapods over here, there's blood vessels that are stemming off of the systemic arteries or the aortas, and they're going to move forward. Instead of them turning around, they're going to move forward and they're going to go in the cranial position because the brain and the face needs oxygen too. And our upper limbs are part of that as well in tetrapods. And so there's oxygen that's needed this direction. And in all cases, going up to the head itself are carotid arteries. Even in fish, there are carotid arteries. They're supplying the head with oxygen-rich blood, slowly moving cranially, and then it's coming back on jugular veins. And again, the carotids and jugulars aren't seen. Coming back either in fish, it comes back on an anterior cardinal, but otherwise they're jugular veins that are oxygen poor returning to the heart. So if you look at the shark, here's the heart over here. You've got the ventricle, and the atrium, and here's the ventricle, and it pumps out through the conus arteriosus through this ventral aorta. So remember, it's on the ventral side, and then it pumps up into these splits. Now, if you recall from dissecting your shark, probably remember that these little splits coming up over here are branchial arteries. And if you really paid attention in lab, and you'll need to know this for a lab exam, so pay attention here if nothing, then they are afferent with an A. Afferent, A comes before E, afferent arterioles. And then when oxygen is picked up in the gills, they're coming out of the gills through efferent with an E, arterioles, and now they're rich in oxygen, and they all converge, they kind of come together until they meet up in the dorsal aorta. I would point out here that going forward is an internal carotid, and then you've got over here anterior cardinal that's draining the head. Over here you've got the aorta is giving rise to all kinds of blood vessels that are nourishing the body, and then it comes back, it's going to kind of come back on veins. I'll get into how things kind of come back later. That's a general idea. The arches, those branchial arteries, are referred to as aortic arches. And the number of arches and how they connect and what they become varies considerably through evolution. So there's major changes in the aortic arches that occur during the evolution, particularly from aquatic to terrestrial organisms. So there's a lot that happens when we have to start breathing air. We already saw big changes in the heart, having to go from a single circuit to a double circuit. And there's changes in the aortic arches that are connected to all of that and to making it all work. Because you can't just pump things out through the heart, they have to get somewhere with blood vessels. So aortic arches are trimmed during development, and some are sort of repurposed and become new blood vessels, like they're there but they're renamed or repurposed. And then once you get from you've got fish, there's some changes that happen in lungfish, and then big changes that happen in tetrapods. After that, between the different types of tetrapods, there are some subtle changes that happen. It's going to be the main focus of today's presentation. I'm also going to talk to you about portal veins. I've mentioned them a little bit in lab, and we'll get into them a little bit more later. But basically, they're just a kind of a set of blood vessels in between blood returning from one organ goes to either the liver or the kidney before it comes back into to the heart. Another big difference in vertebrate circulation just has to do with blood vessels going to limbs. I mean, clearly when something grew legs, you need blood supply to the legs. And so it kind of starts with the existing blood supply in pectoral and pelvic girdles, and then as the limbs evolved and grew longer and so on, then the blood supply went down into those limbs and blood vessels split and became new things. And we're really not going to talk about it much because there's only minor variations between tetrapods. The main focus of today's presentation has to do with these aortic arches. And so the basic pattern of vertebrates begins as 6 pairs of aortic arches that are in embryonic development. And this is sort of a prototypical example, this would be found in agnathans, in those jawless creatures. And so you've got a ventral aorta coming out of the heart that would be poor in oxygen, and then it goes through these arches. And if you see here, one, they start numbering. One is farthest away, it's closest to the head. So 1, 2, 3, 4, 5, 6. 6 is closest to the heart. And as blood goes through these arches, guess what happens to it? You notice it goes from blue over here to red. So what embryonic tissue do you think it comes from, and what structures do you think they're associated with? I'm sure you can make a pretty good guess. By the way, if you want more information just on aortic arches in general, there's a decent website that's up there. It's written it's kind of a biology blog from biology students. Okay, in terms of the agnathan aortic arches, again, you've got ventral arches and they go up here through. If you hadn't guessed, what happens is they're going through the gills so they can get oxygen and then coming back out. And so obviously embryonic tissues, they'd be coming out from those pharyngeal pouches. In agnathan, as I mentioned, there's sort of 6 pairs of these. What are those called in sharks? Remember those names? I just pointed out the names a moment ago. So remember, you connected them. And again, this is just kind of a little study for lab exam hint. There's kind of 2 names for those branches, one before and one after. How these arches evolve. This is a nice little cladogram that shows these branches in shark, teleost, lungfish, amphibian, and so on. And you can see here in shark, they're showing all 6 branches are being used, although some sharks don't have all 6 in use. It gives you a good idea of sort of this ancestral condition. Teleost, you can see wherever there's like a white line, they're showing something is lost, something is not being used. You can see a little bit's trimmed out, but for the most part, it kind of looks a lot like shark with just a little bit less at the top. You get to lungfish, and I would hazard a guess to say it's sort of more similar, a little bit more similar to shark and teleost, but then again, it's kind of in between. It's a little bit of a hybrid. You notice they have lungs off to the side because they're lungfish. Big change happened over here when amphibians started. Because at that point, if you notice the plan, just without looking too closely, you'll notice that amphibians, reptiles, birds, and mammals all have some similarities in the fact that they have the same few that are missing in all of them. And then there's some changes or differences between each of them that are subtle. The big changes between especially kind of like teleost, lungfish, and amphibians. So what I want you to do is, we're kind of moving forward, is think about the advantages those changes have given to tetrapods that has allowed them to be successful on land. Here's just another group of these. They're not set up as a cladogram. You can see this white here is just this basic pattern. And then you've got teleost, and you can see here they've already lost a few. By the way, red is when it's oxygenated, blue is showing deoxygenated blood, and then wherever there's a dotted line is kind of showing that there's a blood vessel or an arch that has lost. What I want you to notice as you go along here is kind of which arches are lost, which ones are retained, and especially why. And I want you to keep those questions in mind as we discuss kind of each group. And I'll show many of these pictures as we talk about the individual groups. Here's another view that shows those arches, but instead of showing them from the kind of a dorsal or ventral view where you're looking at, either looking down or looking up at the blood vessels, this is actually showing the blood vessels from the sagittal view from the side. So you kind of get an idea of what direction they're going in from the side. And if you weren't sure about the hint I asked about the blood vessels in the shark, here they are again. Take a look at that. I'll be talking about a number of these. Some of these I'm not going to get into too much. I will point out things like this nectaris. Those are the mud puppies that are amphibians that retain their gills. You notice that their gill arches are repurposed to go to their external gills. So it's interesting they use those kind of structures for that. But most things, they're kind of a rare exception. What I want you to really focus in on this are what the remaining, what's lost. I mentioned before, but of the stuff that's remaining, how is it repurposed? What do these branchial arteries that are in gills become when fish transition to tetrapods? Real briefly, this looks a lot like the agnathan as a shark. Again, 4 to 6 pairs of aortic arches, depending on the species. And again, you see it's coming through ventral, going through these branchial arches, these aortic arches, and then it comes back out to over here is a pair of dorsal aorta that will then converge together. You notice there are some internal carotids that are kind of spawning forward from the aorta to supply the brain with blood. Some sources say all sharks have 6, but other sharks, some sharks have 5. They may have lost branches 1 or 2. This example is actually showing 1 and 2 are no longer active. So it varies a little bit. Teleos the same way. Teleos, they only have 4 branches, so they have lost the first and the second. Those are the ones closest to the head. So they've lost the 2 farthest away from the heart, and they've kept the 4 closest to the heart. And so those kind of they use the remaining aortic arches to get oxygen from their gills. I do want to point out that from this point forward, not a single thing has the first arch. So everything that's evolved from teleos on is missing a first arch. That's never gone back. I also want to point in teleos, I'm mostly going to be talking about these arches, but this is really important to our discussion. It has to do with the gas bladder. The gas bladder in a lot of fish, it provides buoyancy, and in most fish it's not connected to the outside world. They don't use it to breathe. They're using it for buoyancy. And so how it works is that blood will leave the heart, go through the gills to obtain oxygen, and then after it's out of the gills and it's got oxygen, there'll be a blood vessel that sends it over to the gas bladder. Most of the time, they're showing it here, most of the time it's hooked up really close to that 6th arch. And that's important. I'll get to that later. But what happens is that the oxygen that was picked up in the gills can then be released into the gas bladder. And so the gas bladder contains a really high percentage of oxygen. And that oxygen goes into the gas bladder that, again, just keeps the fish buoyant. And then when the blood supply leaves the gas bladder, it's going to be poor in oxygen, and it's going to kind of returns to the hepatic portal vein, to the liver, and then back to the heart. Gives you an example of a portal vein structure. Why does it matter? Why do you care? Why should you care? Well, we'll talk about that in our next video.